Welcome to, I guess, the third GP Neuro seminar. This is sponsored by the Neural Engineering Center. I'm the co-director. My name is Lena Ting with Garrett Stanley. And hopefully, I'm glad to see so many faces here. If you want to see the whole schedule, go to neuro.gautech.edu and look at the seminars tab, and you can see the whole host of speakers that we have lined up this semester and in the next semester. So today, it's my pleasure to introduce my friend and colleague, Michael Boric. He's an assistant professor in rehabilitation medicine at Emory in the Division of Physical Therapy, and he's also program faculty in biomedical engineering. You'll hear a lot more about his work in terms of brain imaging and brain stimulation in stroke. I'm lucky to have him across the hall from me in the rehab medicine building, and he's been attracting a lot of Georgia Tech undergrads to his lab doing various projects, and we are hopefully will be collaborating more with students in biomedical engineering, neuroscience, and beyond. So I think that Michael presents a very nice intersection between our lack of understanding in basic science as well as in a clinically relevant domain. So thank you, Michael.
All right. Thanks, Leah. Thanks. All right, great. Thanks, everyone, for coming and spending your lunchtime. Of course, it's easier when it's actually provided, the lunch. So I was like, wow, there's a really good turnout. And then I realized there's all these boxes in front of you, which makes a difference. So I know it's not all me. It's caloric related mostly. So I should tell you that I'm not an engineer. I think that's probably pretty clear from my title. And I'm barely even a physical therapist at this point. So I'm not really sure what I am anymore. Although I will say that I think that my interests are at the intersection of these fields. So which means I know a little bit about a lot and not a lot about really anything. So keep that in mind today as we go through this. But really, I'm interested in applying different disciplines and the knowledge base of different disciplines to try and answer complex problems. Of course, I can't do this by myself, hence why I jump at these opportunities to give these types of talks, because many of you know much more about many things than I do in your respective areas. And so I'm always excited to talk about what I'm doing and having you inform it from a different perspective. So I'm really excited, and thank the organizers for inviting me. I will try and go through, of course, every researcher has too many slides, so if we don't finish, it's fine. And I've also tried to hit the middle for the audience, so a lot of you, this will be very review in many parts, and other parts will be brand new. So if you don't understand something, feel free to interrupt me. That doesn't bother me if you need clarification or have a question. Okay? So, and I will probably move around a lot because this is a bit of an awkward room, and otherwise you know which way I'm, a turner, but I can't never do 50-50. I've tried many times in rooms like this and never works. So I'll just walk around instead. So I almost start every talk like this. This is not like this because it's the first time I've shown this image, because this is the new stadium that's going up in downtown Atlanta for the Falcons and, more importantly, the Atlanta United soccer team. So you may ask why I'm showing you this. I'm showing you this to motivate the problem. And so the problem that I and many other researchers are trying to solve is that if you took this new stadium, the Mercedes-Benz Stadium, which is seating capacity about 71,000, if you took that and multiplied it by 95, that would be the number of people currently living in the United States that have experienced a stroke. So these are all the people who've survived after having their stroke. Okay? That's maybe a problem. It wouldn't be a problem, though, if they all recovered. If everyone had a stroke and then they got back to the same level of their functioning that they did before their stroke. Problem is, this is a conservative estimate. Over half of the individuals who've had a stroke have lifelong persistent impairment of activities of daily living. They have some level of disability. So all of my work is motivated by the goal of trying to fill the Mercedes-Benz Stadium instead of 48 times, 0 times. So that's really where I'm coming from with all of this. Okay? And then another sort of organizing principle, this is how we conceptualize in the lab and others conceptualize how these different mechanisms relate to motor recovery after stroke. So we're going to spend most of the time talking about motor recovery after stroke. And really we're talking about this relationship between neuroplasticity, what underlies the formation of new memories, specifically in the motor domain today, how skill learning is the sort of the behavioral manifestation of this memory formation. Remember, we can't really measure memory. We can only measure the performance based on that memory that's made. And in terms of stroke, we're looking at the recovery of function that's related to this skill learning, or relearning in this case, depending on what skill it is. So then, now this seems pretty obvious, but one of the foundational concepts is that you have neuroplastic change that underlies learning. And we've seen this now, that you can actually see both structural and functional change in the brain associated with learning of novel skills. And that's what's shown here. We actually show white matter changes now. We've seen structural changes in the brain with learning new skills. We've shown this in our lab. Many other labs have shown this. You also see in cases where, say, there's a disuse phenomenon. So in this study in particular, there was an immobilization period for people after they had fractured their forearm, and they studied the brain, and they looked at the sensory motor cortices on the contralateral side of the brain, and they actually showed a decrease in cortical thickness after only 2 weeks of arm immobilization. This is sometimes thought of as a potential model and something related to the disuse phenomenon we see in individuals with stroke, that when they've had a stroke, their paretic arm, they tend to ignore it, they tend not to use it, and we now have evidence to show that there are structural and functional changes in the brain, and that's what's shown in this figure. These are some old MR images from my former institution. That was really the first study showing that there's a functional reorganization of the brain after stroke, and that if you train someone to try and promote activation of the ipsilesional hemisphere controlling the paretic arm, that you can actually shift, you can change this activity. You can shift the balance of the activity after stroke. Okay. So really what we know, and this seminal work done in the animal model and squirrel monkeys, showed that if you induce an experimental stroke, you see that there are changes in the area that you stroke. So this was a motor cortex stroke in these squirrel monkeys. And if you just let them recover normally, you have a very different mapping. You have a reorganization where the distal forelimb does not show the same amount of cortical real estate anymore. However, if you provide skilled rehab, you actually prevent that loss. You sort of prevent this maladaptive reorientation. So this was some of the foundational work showing that we can actually, through rehabilitation, change the brain. And that's a key point. So everything that I'm interested in is how we can harness this capacity of being able to change the brain through rehabilitation and also through neurostimulation, which we'll talk about in a moment, to change the brain after stroke. There's more evidence now showing in humans that we see the same thing, that if there's rehabilitation, we can shift these cortical representations. So to kind of sum that up very quickly, we now know that neuroimaging and neurostimulation approaches can be used to characterize neuroplastic change in the brain, and that they both have the potential to facilitate experience-dependent plasticity, and that's what we think is necessary to optimize stroke recovery. Again, getting back to that idea of optimizing these functional outcomes for our individuals. So, again, this should not be any news to anyone in this room, given the audience. But I put it up here so you know where my biases are and where my expertise lies. We work really in non-invasive methods and looking at a level from the network up to the level of the whole nervous system. So we're not looking at a cellular level. We're not even really looking at a synaptic level. So all of our, again, take that with a grain of salt, everything that I'm going to show you is really talking about a systems or network level perspective of neuroplastic change. A lot of my colleagues who work in the cellular molecular level of neuroscience would say that the stimulation stuff that I'm going to show you using transcranial magnetic stimulation, which we say is very focal, it's great. Actually, they would say, oh yeah, it's great, it's like dropping a bomb on the brain. Okay, so again, this is, we come from different fields, and that's good, and that's important, and so I just always like to outline sort of our assumptions that we have. Okay, so the guiding question, I alluded to this already. So how can neuroplastic change in the human brain be adequately imaged and meaningfully modulated non-invasively after injury or disease in stroke? Okay. So for those of you who don't know, we use a form of non-invasive brain stimulation called transcranial magnetic stimulation. Again, this is a painless way to induce evoked activity in the brain in the area of stimulation. This actually happens transsynaptically based on how the magnetic field induces electrical currents in the underlying tissue. We see that the focality, again, going back to that bomb scenario, the focality tends to be, is of course dependent on stimulation intensity, but roughly you can think of it, it's stimulating around a centimeter squared area of cortical tissue, and then the depth of tissue also depends on intensity, but you're really talking about only looking at the primary layers of cortical gray matter in the brain in the area that you're stimulating. So this is a stimulating coil. This is not a Georgia Tech undergrad. Unfortunately, I should have changed the picture, and I should know my audience better, but actually an Emory undergrad, and our postdoc is applying TMS to his motor cortex to try and induce a response. So almost the entire field of what we know about cortical physiology using TMS is related to the motor cortex. And the reason why is because you can evoke a peripheral response. So we can actually get something that we can measure. Again, this technique's been around since the mid-'80s. So in the early days, what we would do is measure an evoked response in a muscle of interest when we stimulated the contralateral motor cortex. And that's what I'm showing you here in figure A. So what we measure is a motor evoked potential, or an MEP. And this would be a peak-to-peak value. You can also measure the latency to get some idea of conduction time from the motor cortex to the target muscle. And you can do some fancy things on looking at where delays might be occurring and why those might be occurring, so on and so forth. Okay, so that's what we call an investigatory form of TMS. But you can also apply TMS in a repetitive or patterned manner. And in this way, you can actually modulate cortical activity. So we can both measure and modulate cortical activity using TMS. It just depends on how you apply it. And depending on what frequency you stimulate, you can either inhibit the activity in the area that you're stimulating over, or you can excite or facilitate the activity. So this is all, again, based primarily on frequency. So getting back to stroke, a lot of our TMS work has informed our understanding of the pathophysiology underlying, or what we think is underlying, this lack of recovery for individuals after stroke. What we see under normal conditions is that you have a balance of excitability, of inhibition between the hemispheres. And this is important for if you want to perform a unimanual task. You need to have this inhibitory drive to suppress mirror movements in the opposing extremity, which is great. And this is, we think, mediated by excitatory synapses transcallosally, that synapse on inhibitory interneurons, and everything's great. Okay? However, after stroke, it seems that because we have a disruption in the balance of activity in the two hemispheres, the ipsilesional hemisphere, the stroke hemisphere, is downregulated, so the excitability is lowered, whereas we see a response in the contralesional hemisphere, the non-stroke affected hemisphere, of activity actually being upregulated, so it's increased excitability. So now we have an asymmetry and imbalance. And this would be called our interhemispheric competition model of stroke recovery, that this imbalance is abnormal and preventing recovery after injury, especially of upper extremity function after stroke. And we've looked at this, and so I'll just map out this paradigm very briefly for you, how we look at this using TMS. So how we've actually measured this phenomenon in the human brain in our patients with stroke. So what you see here is the setup where we have an individual performing a sustained isometric contraction. And what we do is we stimulate the contralateral motor cortex that contains the representation for that muscle. And at the same time, we measure from the ipsilateral side. And so what you see here is the typical contralateral motor evoked potential, what we showed you before, except in this case it's rectified, so it's not a biphasic wave. And then what else we do is we look at the ipsilateral activity. So we have ongoing volitional muscle activity, then we stimulate, and after a period of time, we see a reduction in the volitional activity because of the stimulation which is mediated by these transcallosal projections. So we can get an index, an indirect index of the integrity of this pathway, this transcallosal pathway that we think is an important factor in stroke recovery. And I think that's great, but what's even more interesting and probably more important is that changes in the integrity of this pathway, as shown by abnormal values in this transcallosal inhibition, are actually related to persisting motor impairment in people with chronic stroke. So the more abnormal interhemispheric inhibition you demonstrate, the more impairment you tend to demonstrate in your paretic arm. So we have a nice relationship there between those two metrics. So then the bad news. So I gave you all the great stuff because I'm biased. Yes?
Can you say a quick comment about the damage here, about cell death versus just abnormal network function? I mean, are these stroke patients, what level of damage has occurred?
Yeah, so that's a great question. So that always comes up in this idea of lesion location and lesion severity and so on and so forth. In this particular study, none of the individuals had observable evidence of stroke infarct in the projections between the two motor cortices, but we do know that there are things like Wallerian degeneration that occur distant to the site of the lesion that we think are related to this. In fact, we see some evidence, even though there's not a lesion in these areas, we see some changes in the structural integrity of these pathways that we think is a secondary consequence to the lesion. Yeah, good question. So one of the problems is that we're, and I hope you're thinking this to yourself and you're not saying it, and you're like, well, he's talking about cortical excitability, but all he's measuring are muscles and EMG from muscles. This is quite a discrepancy. And I agree, actually. But we've actually learned a lot about how the brain functions by measuring EMG activity. But it's probably not enough. It's probably insufficient. It's a very rather indirect measure. And one of the things that we think is really problematic is that you're limited in the areas that you can stimulate and evoke a peripheral response. So if you can't evoke a response, how do you know what's going on? So most of our, like I said, most of our understanding of cortical physiology is either related to the motor cortex or the visual cortex, where you stimulate and you can induce phosphine activity. And that's really about what we have. And then the only other way we do it is in paradigms where we think there is a certain area of the brain that's involved in a behavioral task. And if you stimulate that area during the behavioral task, does performance decrease? And so we have what we call a virtual lesion paradigm, where if you stimulate that area and performance is worse, then you have some causal inference to say this area is at least involved. Maybe it's not principally involved, but it's somehow involved in performance of that task. Okay. It's also limited in its ability to talk about what's happening between regions. So we can really only talk about the region that we're stimulating when we're looking at this type of standalone TMS measurement, when we're looking at EMG evoked activity. We can really only say something about the motor cortex or areas that we know from other work that are directly connected, like in the case of transcranial inhibition. So the question is, how can we characterize with TMS this rich network of connections that are both at rest and also modulated during tasks? And then, of course, in stroke, what happens in this situation where someone's cortical spinal tract is disrupted and that we can no longer elicit a motor evoked potential? So how can we understand anything about their cortical physiology if we don't have the wiring intact to be able to measure the response? And in fact, in this person, this is what we get. So you'd like to see a nice response here, a nice MEP. However, we don't get one because the signal can't get there. And in a lot of these cases, that cortex that you're stimulating responds normally, we think, to the stimulation. It's just we don't have the marker to be able to measure. So that moves on to what I'm excited about, and what we do a lot in the lab is trying to combine TMS with neuroimaging approaches. And I'm going to give you a couple examples, and this is a nice conceptualization, a great paper if you're interested in this, from Hartwig-Siedner's group, showing that there's different ways, different flavors of doing this. You can perform stimulation while imaging, which is what we're doing a lot of and what we're the most excited about. I'll get to that in a moment. But what's been done quite a bit now is what's called an offline approach, where you can either image and then stimulate, or stimulate and then image, to try and look at the effects of the stimulation, not just at the site of stimulation. And I'll give you just one example of something we've done. Again, this is just kind of picking and choosing to give you a sense of how these techniques are used. So again, here's that model, that interhemispheric competition model. So what happens is we think we can try and modulate activity either to try and upregulate the downregulated ipsilesional hemisphere, or to try and downregulate the abnormally upregulated contralesional hemisphere. So you can do this in both ways, and like I said, depending on the frequency of stimulation, that will determine which response you get. So a nice example of how we see this functional change is that when you have an affected hand, in healthy subjects there's this nice lateralization of activity. This is bold signal. And you see this lateralized to the side controlling the hand that's active. Whereas in individuals with stroke, you tend to see this loss of lateralization, that both hemispheres seem to be activated. And this is some of the more correlative or indirect evidence showing that you have this abnormal contralateral activation when it shouldn't be there. Okay? And you don't tend to see this when they're moving the unaffected hand. So this is specific to when they're moving their paretic extremity, so the hand that's been damaged by the stroke. So we thought, well, could we actually upregulate activity in the damaged hemisphere, in the ipsilesional hemisphere? And this is a study we did a few years ago in the lab that I postdoc. And we tried to excite the area before training, so before rehabilitation, to see if we could prime the system to learn. That's the underlying theoretical grounds for this. So we stimulate them for a period of time, and then we have them engage in a lab-based motor skill task. And what do we see after 5 sessions of doing this? We actually see that they get better compared to sham stimulation. So just practicing the task, they get a bit better. But if you then do priming repetitive TMS, if you modulate the system before they train, we actually see greater benefits. That's okay, great. Problem is that not everyone shows this, and this is the sort of the bane of the neuromodulation community, is that some people respond with a very pronounced change in excitability and behavior, and other people show nothing. And this is especially problematic in clinical trials in stroke, looking at neuromodulation, because what do you see if you go across a large group? This is what you see, and you group average these data, you don't get a lot in many cases. So we wanted to know, are there biomarkers of response to stimulation? So we imaged them. We imaged them before they started the study, and then we looked at the data retrospectively. And what we did is we ran and looked at volumes of cortical tissue directly beneath the site that we stimulated, in this case, the somatosensory cortex. And what we saw is that cortical gray and white matter volume predicted about 72% of the variance in their response to this pairing of stimulation with training. And we didn't see any relationship in the group that had the sham stimulation. So this seems to be specific to the response to the stimulation, not the response to the training. Does that make sense? So this is one example of how we may be able to better understand responses to neuromodulation by getting imaging information about these individuals. And for example, you could think about using this to say, oh, you show this level of volume, white or gray matter volume beneath the area that we want to stimulate, you're likely a good candidate for stimulation. Or you show this level, you're not a good candidate, let's try something else. And so this is a way to try and tailor or individualize neuromodulation. And we're trying to do this with many other treatments as well. I'm just keeping it to neuromodulation at this point. We think it's related to this idea of secondary degeneration, but we don't know. So that's the other sort of elephant in the room, is that we don't have imaging on these individuals before they had their stroke. And we don't usually have it right after their stroke, but it appears to be in some other work that it is this process that you see degradation secondary to the site of stroke, and probably related to disuse. Yeah. Yeah, Lee?
Do you think if people have greater thickness just because they are more adept, that would deteriorate less?
Yeah, so this idea. The answer is probably, but we don't know, and that's the other question. There's some work actually by Bruce Creston's group over at the VA and at Emory showing that a lot of the things that we're seeing in our individuals with stroke maybe aren't because they had a stroke, but because they're old. And when you get older, you tend to be more likely to have a stroke. So all of this I may be telling you today is because these people got old. I'm not ruling that out. We don't have a lot of good data about that. So you guys are the smart people in the room, figure out how to stop aging, and we probably figure out how to stop most of this. And then I don't have to work anymore. It's great. Okay, so now moving on to what I'm really, I think, excited about, and what our lab's really excited about, is this idea of combining stimulation with imaging in real time. So rather than imaging them before or after, why not image them while we're stimulating to get a real-time estimate of when I stimulate here, what happens in that area that I stimulate. Okay, great, that's the kind of the information that we get, except now a more direct measurement of that. Or, more importantly probably, what happens over here? What happens here? What happens here? And you can, this is a very nice hypothesis generating design where you can get, again, these causal inferences based on the stimulus response behavior of the system. We're using EEG to do this, so electroencephalography. Very briefly, how does it work? Really, we're looking at scalp recordings of potential changes based on stimulation, although you can do this at rest, of course, and you can get a lot of good information about how the brain is behaving just without the stimulation. However, we're going to focus on stimulation. And again, in this case, we're talking about populations of coherent postsynaptic potential activity. So not looking at direct action potential responses, but looking at sort of a coherent activity of these neurons that we're stimulating over, and then the neurons that are connected in between. And one of the important things is that we get a temporal resolution of about a millisecond or better, which happens to be quite nice for biologically relevant timescales. So although our spatial resolution is maybe not as good as some of the molecular approaches, the ability for us to look at timing of responses and timing of signal propagation through a network is actually really on line with what we might expect in biological processing. So here's an example. What we do is we actually have fancy software that we can identify and target specific areas of brain that we're interested in. So we're no longer now required to be able to evoke a peripheral response to say exactly where we want to stimulate. We can use an individual's own MRI data, so we can look at their structural brain data and say we want to target here, or we want to target there. For example, if they have a stroke here, maybe we want to target the paralesional, so the area surrounding the stroke. We know that area is more plastic, especially in the early stages after stroke, so maybe that's the we want to target an individualized stimulation for a given participant. Okay. And then what we do is we stimulate and we actually record responses to the stimulation. So at the y-axis here is when the stimulation is delivered, and you can see different evoked potentials that occur in the channels that we're recording. We record 32 channels of data in our lab. You can do more, you can do less. This is what we do. Okay. And so this is what you see. So these are just a kind of a toy example of what we get. So I imagine that many of you are thinking, wow, you stimulate, you get about a 2 Tesla magnetic field in the area. That's probably not great for, you know, measuring microvolt potentials. It's true, it's not great, actually. But we've actually advanced the hardware to an extent where the artifact, which is what you see here, time point 0 is when the stimulation is delivered, and you see a large artifact that I've cut off here so we can actually look at the EEG data. We get about a 20 millivolt response around the peristimulus interval. But what you see is that we actually get resolution of that artifact within about 5 milliseconds of stimulation. So after 5 milliseconds, so we lose that data. Sorry, it's gone. Okay. But then after about 5 milliseconds, we get clean, relatively clean EEG data that we can actually interpret that's not affected by the TMS artifact itself. Okay, and that's what you're seeing here. And you can see how the signal, again, the distribution of the spatial information that's represented by the signal over time. And you can see it starts laterally, this is left hemisphere stimulation, and over time it propagates contralaterally and moves throughout this system. So again, this is just sort of an illustration of what kind of information, the richness of information in terms of spatial and temporal data that we can get from this technique. Again, compare that now back to an MEP, which you saw we can measure the peak to peak in one or a couple channels and how long it took to get there. So this is a substantial increase in the amount of information that we have available to us. The problem is, what do you do with it? In fact, we've done the simplest thing we could possibly think to do in our people with stroke. We said, okay, we have all of that information, but we're kind of, you know, new to this. So why don't we look, and we don't know in people with stroke, this hasn't been done. So what do we see when we just look at one EEG channel and we evoke responses? And what we see is that the actual responses that we get in people with stroke are different than those individuals who are matched for age and gender. So this, in this case, we wouldn't attribute solely to aging. This is a response related to the stroke. Not saying there's not some interaction between those two things, but we think at least because we're matching to our control group, we have some better indication of what are stroke-related effects compared to just aging in general. And what we also see is that one component of these responses, so again, not only in the MEP we get one response, one evoked response, in these cases we actually get a response that has multiple peaks to it that we think, and data have shown, some pharmacological data, that are related to inhibitory and excitatory circuitry, that we can actually get some estimate of the integrity of those different circuits and those different pathways, depending on which of these peaks is altered or abnormal after stroke. Okay, so that still doesn't really tell us anything about connectivity or how this network approach, right? I just looked at one channel and said, okay, these are different. So that's nice, that's really clinically feasible. We could do that very fast. We could get some estimate potentially of the brain activity in an individual with stroke. Maybe it helps us with our decision making, maybe not, but that's its advantage. Its disadvantage is it's, again, not capitalizing on the richness of all this information that we're collecting in real time. You know, this idea that we can actually get information about resting or task-based networks using this approach. So that's what we're trying to do. And you can measure this, of course, in the time domain, or you could look at this in the frequency domain, which is going to be important soon. So I just wanted to show you, and some of you maybe who've been to another talk of mine have seen this before. Let's make sure we get on the right. So this is an example. I'm just going to walk you through and set up this video before I show it. This is a top-down view of the brain. It's just a stylized version. And what you're going to see is sort of a graph theoretical approach that some of my colleagues have developed to look at what happens and look at the spatiotemporal dynamics. Now, in the frequency domain, looking in the alpha band frequency domain, and looking at measures of directed connectivity. So in this case, we're going to be stimulating the right motor cortex at time point 0. And what you'll see is changes in the activity in these areas. So we basically have these nodes and edges in speak that you guys might be more comfortable with. And what you'll see is that there's this huge induction of activity throughout the pathway. And what you'll see is, oh, this looks like it's going to be very hard to understand. And I think you're right. But I think someone said at the last talk I gave with this, maybe you, Garrett, was good luck, was what was stated. So this is before. So again, these areas are connected before the stimulation happens. And then now stimulation occurs. We see a large increase in the activity in the area that we stimulated, but also throughout this complex network. We're still, of course, early days in trying to figure out how this network works and what are the kind of the salient nodes of this network. Does this network change when they're active versus they're at rest? Is it different in people with stroke? This happens to be in a healthy individual. How does this network look in someone who has damage? So on and so forth. The point is to motivate what the technique could potentially tell us and how we can. Now, again, the key here is that we're non-invasively looking at these responses, and we're perturbing a certain node in the system and seeing how the areas that are connected to that node respond. Yeah?
How did you identify the nodes here?
Yeah, in this case, that's a good question. So we took the EEG data and we decomposed it into source level activity, and then we projected those sources in standard space and used those as our nodes of activity. So it's a purely data-driven approach in this case. And that's one of the limitations, is that we only have 32 channels of EEG data, so we're probably underestimating the number of nodes that we're able to record. But
Is there an advantage to doing the source localization and not just using the electrode sites themselves?
Good question, and we'll kind of go into that a little bit more in some of the other data we've been looking at and looking at simple measures of coherence. So one of the challenges here is understanding connectivity when you're looking at signals that are a mixture that are receivers from multiple areas of the brain. So that's the limitation, this idea of volume conduction for scalp EEG. So these sensors are picking up from everywhere the entire time, and there may be some bias to local signals in the area, but really you're picking up a mixture of signal. So there's this idea of sort of autocorrelation that could occur between two channels that are close to one another, that they're not actually correlated activity, they're just picking up activity from the same source. And so that's really what motivated this approach, is that we avoid some of those issues. Of course, there are other issues with trying to localize where these sources are and so on and so forth, but that was the rationale in this case. Thanks. So why do we care about brain oscillations? Make sure we're doing okay. So we know that this activity is highly coordinated, and we think that it's quite important for information processing and signaling between areas of the brain, and that we also know for our interest, I don't know which, okay. It's modulated by behavior and learning, and that it's controlled by inhibitory synaptic transmission, which will be important in the subsequent slides. And we've also now have evidence from behavioral data that it's abnormal after stroke. We see abnormal oscillatory activity in sensory motor networks after people have had a stroke. So these are all motivating reasons why we might be interested in evaluating oscillatory activity in the brain. And so we're kind of running a little short on time. I want to show you a few things additionally. But what we're looking at is coherency as our primary metric in this particular case, to look at the activity that's related in the frequency domain. We think of this as sort of a correlation, but now in frequency rather than in time. And again, the challenges that we have related to this volume conduction and that you could have autocorrelation, we think have been solved. I won't go into the gory details, but there's, and in fact, if we want to go into the gory details, Lewis is over here too, who's been collaborating with me on this project and looking at how we can minimize this potential that we're actually seeing real relationships between the activity that we're measuring. So we want to look at in people with stroke, does this coherence between the two channels lying, so C3 and C4 in this case, the two channels that we think are primarily overlying the motor cortical regions of the brain. And do these change? Again, with the hypothesis being we've seen now through TMS measures, through functional MRI, that the interactions between these areas are abnormal after stroke. So can we get some index using this TMS-evoked measure, a more direct perturb and measure approach? Can we also see some evidence of this phenomenon? So now this is without EEG here. This is looking at that TCI measure I showed you earlier where they're contracting and we stimulate and seeing how much inhibition we get in that volitional activity. And we see that in individuals with stroke, there's less evoked inhibition when you stimulate the ipsilesional hemisphere. So we see less inhibition, this going along with this idea of being down-regulated. So we see that with our TMS data, so this replicates some of our data we have from the past, which is always good. But what we see looking at primarily in the beta frequency band, an actual increase in this coherence between these two channels, C3 and C4, which would suggest a greater coupling between the two areas. At first we thought this might be the opposite of what we would expect, but then looking back and thinking about that fMRI literature, again, this is related to when they're actually performing a volitional contraction, we see a tighter coupling between the two hemispheres, which we would not expect to see based on our control data. So these are now evoked responses that show abnormal coherence in the, what we think, motor-related band of EEG. What's also very interesting is that this is specific to motor state. We don't see any abnormalities in this measure of interhemispheric coherence when the participants were at rest. We only see it when they're actually engaged in a motor task. So this is a movement-related phenomenon, not a pervasive global phenomenon. Okay. So what we've seen also, again, most importantly for us, is that these abnormal interactions are actually related to the severity of motor impairment. So the more severely impaired you were at the chronic stage of recovery, the greater the coherence that we saw in our measure. So this idea that more abnormal interhemispheric coherence is more related to severity of arm impairment after stroke. So then that now leads us to today and actually tomorrow. So I have no problem showing data that's new, and so we're going to show new data for the rest of the time so you can tell me what you like and don't like about it. So the question is this a potential biomarker that we could target? Is this something that we can we move the needle? So we've basically taken a cross-sectional and said this is different, and it appears to be related to impairment, which is what we ultimately want to change. So now can we actually change it? So the flavor of neuromodulation that we use in the lab is called paired-associative stimulation. Some may be familiar with this, some might know it by another name, but essentially we're trying to, at least we think, capitalize on the phenomenon of spike timing-dependent plasticity. In the traditional or classical paradigm, the stimulation of a peripheral nerve, in this case the median nerve, is paired at a certain latency with contralateral stimulation of the motor cortex to try and get a synergistic firing. So this idea of presynaptic activity in close succession to postsynaptic activity. Again, this is on a systems level, not at the level of the synapse, of course. But that's the phenomenon, at least, we're trying to replicate. And what's been shown in many, many studies is that if you repeatedly pair these two stimuli at a specific interstimulus interval so that these two pulses coincide rather than be at different timings, you actually get an increase in the responsiveness of the excitability in the area that you stimulate. And this is important that we can show that this is somewhat, or we think, is a human brain analog of what we see in vitro looking at cell preparation, is that we see the idea that it maintains these characteristics, that it evolves quickly, so it takes less than 10 minutes to induce this change in excitability, that it's reversible. So if you look at this person 2 hours after they've been stimulated, the excitability levels go back to baseline before the stimulation. However, it does persist beyond the period of stimulation. So if we stimulate for 10 minutes, we tend to see elevated excitability for about 60 minutes after the stimulation period is over. And it follows temporal rules. So like I said before, if you change the stimulation, so in this case, they did an interstimulus interval where they applied the stimulation peripherally 25 milliseconds before they applied the central pulse. If you change that to 10 milliseconds, you actually get a reduction in excitability. So what's thought to be an LTD-like analog of plasticity if you change this and have them asynchronously arrive at that motor cortex site. So we can debate this and those who think this is garbage and so on and so forth, but that's a whole other conversation. But that's what the field is arguing, and there's been pharmacological data to show non-invasively that this is modulated if you provide, say, GABA agonist and antagonist. You can change the responses that you get. So we've done this in the lab. And we're sort of the first ones to use this approach, this specific sort of traditional approach in individuals with stroke. There's a small cohort so far. What we see is when we provide the active stimulation, where we're stimulating at that interstimulus interval of 25 milliseconds, we see an elevation in excitability in these individuals that persists to about 60 minutes after stimulation. You might look at this and see these standard deviations are quite large. That goes back to greater issue of who responds and who doesn't. We tend to see some of our individuals respond quite a lot to this stimulation. They show a large increase in excitability, and others show no increase. We're still figuring out why that is, but that's some of these data I think will inform that. But in the control condition, where we essentially apply the same peripheral stimulation, but we apply a sham TMS stimulation, so there's no pairing, there's no coupling of these two stimuli, we see essentially no change in the responses after the stimulation period. Okay. So the question is, can we do this directly though? Because again, if you remember back to that image of the diffusion tensor image that I showed you with a disrupted cortical spinal tract, well, this is not going to probably work very well for that individual because you can't pair two stimuli if they can't get there. So are there ways that we could actually look at cortical active networks and do the same thing? And this has actually been shown in healthy individuals where you can actually pair stimuli at certain interstimulus intervals based on the signal conduction time between the areas, and you can actually induce these analogs of a neuroplastic change, and they're also dependent on the timing. So that's what's shown here in this particular example. And it turns out in healthy individuals you do the same thing if you pair stimuli to the two motor hemispheres. So if you condition one hemisphere at a certain latency before the other, you can actually increase the excitability of that hemisphere, which is shown here as there's an increase, but you also decrease the amount of inhibition that you have between the hemispheres. So you can modulate the interhemispheric inhibition that I was mentioning before is abnormal after stroke. So the question is, can we modulate this inhibition in individuals with stroke? We've shown that we can do this in the lab in healthy individuals. We show very similar results to what's been published. Again, there's only one group that's ever done this, so we weren't quite sure that it actually was real, and we weren't actually sure that we could do it, but it turns out we can, and it does appear to be real. So now we're looking at in prime time where we have an upcoming clinical trial which we're starting this fall. So I wanted to, in transparency, show you sort of what we're aiming to do. What we'll do is we'll look at collecting some imaging data on them so we can identify some potential biomarkers of response to this intervention. Like I said, not everyone's going to respond, but how can we then use that information to inform subsequent studies? And also assessments of motor behavior. So is it that individuals that are more impaired, they show a greater response, they show a lesser response? What might we be looking at? And then in the given next visits, we'll do this active or control inhibitory pass. So we'll either have them get the stimulation where we expect that the two stimuli will arrive coincidentally, or we'll have them arrive asynchronously and we expect nothing to happen, or an actual reduction in response. And then we'll look at measures of cortical excitability and measures of motor behavior, both before and right after the stimulation, up to 60 minutes. And then importantly, so again, really, this is not that interesting to you all, but interesting to me in the field, is that very few studies looking at this paired associative stimulation to induce neuroplastic change, very few studies, if any, have looked at what's the ability to learn. What effect does it have on learning? It definitely shows an effect on motor performance. We've shown that now. But there's almost no evidence showing that it actually induces changes in learning, which are necessary, of course, if we're going to induce long-term changes in behavior. Okay, so that's another important aspect that we're looking at, just a simple 24-hour retention phase. Okay, and these I won't bore you with the primary outcome measures since we're running short on time, but this is, suffice to say, what we're looking at in terms of our basic measures of motor skill performance in the lab, and also looking at more functional measures of our individuals and seeing how their paretic arm function may change as a result of this neuromodulation stimulation paradigm. Okay, so we're to the end. You've hung with me. I appreciate it. The lights are low. The food's working. It's magic. So thank you for that. Again, I hope I've convinced you that neuroplastic change occurs across the lifespan. I hope you maybe were convinced of that before you walked in here, but we've sort of reinforced that. That TMS can both measure and modulate this neuroplastic change. And now we have the ability to directly evaluate this ability to evoke neuroplastic change using combined TMS imaging methods. The one I showed you today was TMS combined with EEG to try and give us more direct estimates of what we're actually doing to the brain as a result of these neuromodulation paradigms. So with that, I'd just like to acknowledge my collaborators as well as all the individuals who are helping collect these data and work on these projects. And then I realized before I walked in today that this is mostly meant to be a space to share research about pediatrics. And I don't have anything about pediatrics in my talk, so I went back real quick and I put in a slide from my past days as a postdoc where we actually did look at structural biomarkers of sport-related concussion in adolescents. So we actually showed that if you do diffusion imaging, you can actually see changes in the brain after a sport-related concussion in these adolescents that far outlast the resolution of behavioral symptoms. So I thought I would just throw in one of those. And also, if you want to talk to me about structural imaging, we do a lot of that, but there's just not time to talk about all of it. So with that, thank you, and I'd be happy to take any questions, and yeah, appreciate it. Yeah.
Has anyone done animal studies on the paired stimulation paradigm? It seems like it would be a really easy thing to do. You could look at synaptic change and all sorts of things.
Yeah, not really. Actually, in terms of looking at TMS as well, they've just really gotten to the point where you can actually have a coil that's animal size, so to speak. But to my knowledge, at this point, I don't know anyone who's actually done that study. But yes, I think it's quite an easy study. There's been other studies that indirectly have looked at, evoking responses that slightly different way, but not pairing them with TMS itself, because of course the mechanism is slightly different. But there's been some work doing, related to, looking at trying to stimulate using light, infrared. I'm blanking on the paradigm, but that's been done a little bit, but it's slightly different in terms of the mechanism. But yes, that, to my knowledge, hasn't been done. Yeah. Good question. Yeah, too.
Thanks, Michael. That posed a lot of questions. Try to be brief. One is, I guess, we thought about stimulation having an effect not by direct activation of neurons, but by activating BDNF or some kind of secondary sort of signaling pathway that produces plasticity.
Yeah, so we have thought about that, and some other individuals have looked at that, and we, in my former lab, we were collecting some samples, saliva samples, and looking at, does BDNF tell us anything about response to the stimulation? Because we think that it may be, it's obviously related to neuroplastic change, but is it also related to response to the stimulation? It's a bit muddy, I will say, with that. But in terms of just response to the actual stimulation itself and what's causing that, there's pretty good data now showing that the responses are mediated by these transsynaptic connections, but we don't know how much of these secondary messenger pathways or BDNF are really functioning there. Most of the pharmacological data is really looked at, GABA and glutamate and how those particular neurotransmitters are, working. Yeah.
Just looking at the ipsilesional side, do you see anything in your data that would help explain unwanted synergies?
In our data, no, we haven't particularly looked at that, but that's something I think we've talked about a little bit and potentially looking at the synergies. In fact, one of the problems or one of the limitations in our setup is that we don't collect enough EMG channels to look at that data specifically. I think some other groups have looked a little bit at synergy and how it relates to response to stimulation, but we haven't done that specifically. But I think it's a really good question and something, as terms of talking about responses and what might be changing, because again, I showed you behavioral data, but really those data are talking not about quality of movement, they're just talking about absolute results or performance. So it doesn't tell us anything about, are they moving differently, are they moving better, are they decreasing their number of synergies when they perform these activities? We don't know very much about that at all at this point.
And if I just limit
Yeah.
myself to 3.
Yeah, that's good.
Are people trying to validate these measures in non-human animal experiments?
Yes. And in fact, Catherine Butefish is doing this right now in a rodent model, trying to replicate the TMS data. And we have a collaboration back at UBC where we're doing it in a primate model. And then others have done similar things, yeah.
The individual differences in response
For the initial motor cortex or motor cortical excitability, is there a correlation to how responsive they are to the cortico-cortical?
That's a very good question. The short answer is we don't know because it hasn't been done, but I'm trying to think of what I might hypothesize is in that case. And I think so because if we just look at the excitability data, they tend to correlate that if you see reduced ipsilesional excitability, you also tend to see a reduced interhemispheric inhibition signal as well. So I would expect that if those two pathways would be modulated similarly. Yeah, partially because of the lack of specificity to specific circuits with the stimulation. So those are mediated by different circuits, but we don't have the ability to target uniquely each of those circuits. There's ways we can play with that, but it's not perfect, of course. Yeah. Oh, sorry. Yeah. Oh, sorry.
I want to ask you about the pair-associative stimulation using 8 millisecond difference. This seems to be like a magic number that everybody's using. Has it been validated with like different? Has anybody plotted how different
intervals, interstimulus intervals. Yeah, so that magic number primarily came from early data back in the 90s. They did a similar paradigm and showed they did basically a ISI response curve and looked at what was the optimal interstimulus interval that evoked the largest inhibition between hemispheres. And it seems to be 8 to 10 milliseconds is sort of the sweet spot. Yes, but then, of course, and then anything outside of that, now you're talking about indirect pathways that might be related. If you extend that interstimulus interval, which we're also interested in, but we haven't gotten to that yet.
Yeah. There was actually a very recent study showing V1 to MT. They used 20 milliseconds and they got the same result. So that was very surprising to me.
Yeah.
Physiologically, it's actually a big difference.
Yeah, and we think maybe some of our TMS-EEG data can shed some light on that. As we mentioned before, it's really capturing this, these postsynaptic potentials, which are on the tens of milliseconds rather than on the millisecond in terms of their activity. So it might be related in that respect to some of these timings of these different pathways and looking at that. So rather than a direct connection, maybe it's a secondary activation, kind of getting back to Tim's point and what's actually happening here. Again, to be quite honest, that's the challenge of doing the non-invasive human brain stuff is a lot of this, my hands start to reflexively start waving a little bit. I apologize for that, but that's, these are the questions that I'm really interested in answering, that we've hand waved quite a lot in the past, and now I'm trying to have that amplitude of waving get smaller and smaller. Yes. Yeah. I'll be quick too.
So in looking at the spatiotemporal dynamics in the cortical network, is there a role for models like the virtual brain to actually constrain what's directly related to the stim? Because we know there's a speed limit, and anything that looks like it's traveling faster than the speed limit is probably not related to the stim.
Exactly.
Is there work kind of being done?
I don't know if there's work being done, but that's some of the ways that we're looking at how can we constrain these models. And so what I showed you is really modeling the propagation of activity through the network, and that's the constraints that are put on that model are certainly open to playing with in terms of that. And using, like you say, already understood rules will inform that, I think, to a greater extent. We just haven't had the chance to really dig our teeth in and be very sophisticated about it quite yet. It's a good question. Thank you.
Great, thank you.
Yeah, thanks, Lynn.
Nicole. Let's take our speaker again.
Thank you.